All right, can you see my slides?
Yep, and I can hear you, although it's a little quiet. So if you've got a microphone or something, you might want to move that closer. But yeah, take it away.
All right, thanks for coming to my talk. I'll be talking about the predicting fate of nitrogen during spider-arthropod trophic interactions. So food webs are, they show how energy and nutrients move through ecosystems, and at each node of a food web, resources are transferred from a food item to a consumer, like prey nutrients to a predator. And then some of those nutrients are assimilated by the consumer and some are deposited as detritus. Spiders are important members of food webs. They're abundant. They consume a large amount of prey in a diversity of different prey. And so they form this important link between lower trophic levels and other types of predators. I've been interested in understanding how spiders affect the flow of nutrients through food webs and ecosystems. In the past, in my lab, we've used feeding experiments to look at the nutrients from prey that end up in predators. So basically, we feed a prey item to a spider, and we look at what's ingested by the spider and what's left behind as prey remains. And we can calculate ingestion, assimilation, excretion, and prey remaining. So it gives us really great data on the transfer of nutrients at a single trophic interaction. But the only problem is that we can't, it's not feasible to do these type of studies with all potential prey items. There are many diverse prey that spiders can feed on. And so I've been interested in seeing if there are traits of prey items, like things that we can measure in whole prey items that will predict how much of the prey nutrients will be ingested by a spider versus discarded and end up as detritus. So like I said, we've done a bunch of feeding experiments where we take whole prey, live prey, we feed them to spiders, and then we measure what's left behind and what the spider consumes. Here we have two examples of prey. We have a mealworm beetle and a cricket, what the whole prey looks like and what the prey remains look like. And what we see through these studies is that for some prey, there are a large amount of prey remains. The spiders consume less, and for other prey, there are much less prey remains, and the spider consumes relatively more of the prey. So I've been interested in understanding if there's something about these whole prey that we can measure that lets us predict how much will be left behind versus eaten by the spider. Now, if we look in detail at these prey remains, these are on the right are two pictures that I've taken from studies, and on the left are images from the Biology of Spiders book. We look at the prey remains, they look like they're mostly exoskeleton. So just pieces of exoskeleton that in some cases look really clean of any other tissue. And so the goal of this work was to see if measures of exoskeleton content of whole prey can help us predict how much of that prey will be digested by predators versus left in the environment as detritus. So to do this, I compared what spiders ate from a variety of prey. So we did feeding assays on a variety of prey, measured spider consumption, and then we also measured whole prey and tested what was a better predictor of spider consumption, just the whole prey mass or metabolizable mass or nutrients. And for metabolizable mass or nutrients, I calculated that as the whole prey minus the exoskeleton. So basically, the parts of the prey that weren't exoskeleton, does that predict what the spiders are eating better than just the whole prey? So for the methods, we used field collected prey, 22 different morphospecies from 16 families, included arachnids and insects. Again, we measured what was in whole prey items, dry mass, exoskeleton, carbon, nitrogen. Carbon and nitrogen because they're often the currencies that are used to study food web and nutrient dynamics in ecosystems. Then we fed these prey to wolf spiders and measured what the spiders ate and what the spiders left behind. So the first data I want to show are percent consumption by the spiders. So this is a histogram showing the number of prey and percent consumption of those different prey. So we can see that spiders consume between 45 and 90% of prey mass, and that's a huge amount of variation. For some prey, they consumed less than half of the dry mass, and other prey, they consumed nearly everything. And the species were pretty widely spread within this range. And so there's a lot of variation in how much spiders are eating of individual prey items. Next, I wanted to use the data on exoskeleton content to see if there was a relationship between this percent consumption and the exoskeleton content of prey. So we have three figures here, and all the figures, percent exoskeleton is on the x-axis, and then consumption is on the y-axis. So here we have dry mass, the percent dry mass consumed from a prey relative to exoskeleton. There's a significant negative relationship. So when prey have higher exoskeleton content, spiders consume less of the dry mass. And that makes sense. This exoskeleton, we believe, is indigestible by the spiders. And so if there's a lot of exoskeleton, the spiders are consuming less tissue from the prey. We get the same sort of relationships for carbon and nitrogen. When there's a lot of exoskeleton in the prey, spiders consume less nitrogen from that prey and less carbon from that prey. So basically, less of that prey material is digested. And what's really interesting about these figures is that these are field caught prey that vary in all sorts of characteristics. We didn't measure any defensive compounds. We just measured exoskeleton content, and that's explaining quite a bit of the consumption, percent consumption of these prey. So a very simple measure explaining a lot of variation in prey consumption. Next, we wanted to use these, see if metabolizable or total nutrients were better predictors of consumption. And so here we have in the blue line, so on the y-axis, we have dry mass consumed, and on the x-axis, we either have the total dry mass present or the metabolizable dry mass present. And again, metabolizable dry mass is just total dry mass minus what was in the exoskeleton. So presumably, metabolizable is what is non-exoskeleton, what should be digestible by spiders. The blue line is a relationship between total dry mass and dry mass consumed. The orange line is a relationship between metabolizable dry mass of a whole prey and dry mass consumed. We can see that the metabolizable measure of whole prey is much closer to a one-to-one relationship than what's present in the whole prey body. We get the same sort of relationship for nitrogen. So again, nitrogen consumption is on the y-axis, and either total nitrogen present or metabolizable nitrogen is on the y-axis. The blue line is total nutrient present. Orange is metabolizable nitrogen content. And we can see that the metabolizable nitrogen content of a prey is a better predictor of what spiders eat than just the whole total nitrogen content of that prey. And then we looked lastly at carbon. So again, total carbon present is the blue line. Carbon present in the metabolizable portion of the prey, the non-exoskeleton portion of the prey, is the orange line. And again, the metabolizable content of a whole prey item is a better predictor of what spiders consume when they feed on that prey item than just the total nutrients present in that prey. So for conclusions, the exoskeleton content of prey is inversely related to how much of the prey that the spider eats. So when prey have high exoskeleton content, spiders consume less total dry mass, nitrogen, and carbon from those prey items. This exoskeleton really varies widely among prey species. So in our data set, exoskeleton varied from 14 to 44%. So there's a lot of variation in this exoskeleton. The exoskeleton is composed of chitin and proteins. A lot of times when people think about exoskeleton, they think just of chitin, which has very low nitrogen content. But there are a lot of proteins that are woven within the chitin, and those proteins within the exoskeleton are indigestible to predators, but they increase the nitrogen content of the exoskeleton. And so some of our data shows the exoskeleton of insects and spiders is about 10.3% nitrogen. which is similar to the nitrogen content of whole arthropods. So there can be quite a high concentration of nitrogen in that exoskeleton, and it can be quite a large percentage of the prey mass. And then our data suggests that this metabolizable nutrient content might better inform trophic interactions. It can tell us what parts of the prey are likely to be eaten versus discarded. And so simply measuring this exoskeleton content, which is, it's very easy to measure. Basically, you take a dry prey, soak it in sodium hydroxide, and let the sodium hydroxide dissolve the soft tissue. And then what's left over is exoskeleton content. So instead of measuring a whole prey, separating it into metabolizable versus exoskeleton can give us more information on the fates of nutrients, where those nutrients end up in food chains. So if we think about potential implications for food webs, if a spider is feeding on a low exoskeleton prey, a relatively higher proportion of that prey gets transferred to the spider, and less of that prey ends up as exoskeleton deposited on the soil. If spiders are feeding on a higher exoskeleton prey, relatively less of the prey ends up in the spider, and more of the prey ends up as exoskeleton in the soil. And this exoskeleton likely decomposes very slowly. And so we think about situations in the field, if there are shifts in the community composition of prey towards lower exoskeleton prey communities versus high exoskeleton prey communities, that could affect the relative amount of nutrients that move up the food web versus are deposited as detritus in that ecosystem. And we're continuing some of the work looking at other implications of exoskeleton for decomposition and looking at spider excretia and how that affects soil and plant growth. So some of the other ecosystem properties related to these nutrients. With that, I'd like to thank a colleague for elemental analysis and my lab group and funding from NSF. Thank you.
And thank you. These are the pictures I was hoping for too. So, okay, Shawn, you've got a question right away from Mike Draney, who asked, he points out that many spiders don't masticate their prey. Have you looked at ingestion in any of those spiders?
Yeah, so we haven't directly compared spiders that masticate versus don't masticate, but we've done experiments with black widow spiders and wolf spiders. And in both cases, when I measure the prey remains, I typically measure lipid and protein content. Both the black widow and the wolf spiders seem to extract about 90 to 95% of the lipid and protein from the prey. So it'd be interesting to directly compare them in the future, but it doesn't seem like there are any, from what we know now, it doesn't seem like there are any major differences between them.
You've got two questions that popped up. I don't know. Peter, is it okay because he might be able to answer these together?
Yes, it's okay.
It's okay?
Track, yeah.
Okay, all right, good. So we're at 2:45, but I am so good as a moderator. I'm going to give you extra questions. Blaine Hebert asks, how did you determine carbon and nitrogen content? And actually, Maddie Hanapple asked a very related question, which is, would this affect interpreting stable isotope data for carbon and nitrogen for estimating diets in spiders? So perhaps you can answer both of those questions in one fell swoop.
Yeah, for the carbon and nitrogen, we used an elementar. So basically you take a dry prey and grind it into a powder and then put it into the machine, and it measures carbon and nitrogen content. These results, I don't know what it might mean for stable isotope data. It'd be really interesting to look at stable isotope concentrations in the soft tissue versus the exoskeleton. I don't know if they would be different or not, but that's a really interesting question, and definitely would be interesting to follow up on. Because yeah, it could have important implications, especially for some of the prey like beetles and some of the Hymenoptera that have very high exoskeleton content. If the fractionation is different for the exoskeleton versus soft tissue, that could really affect isotope models.
Okay, thank you. Yeah, Peter, how are we doing on time? Is it okay to move to the next talk, or should we still delay a bit?
I have not seen anything from the other track that they are to their questions yet, so we can take another question or two.
Okay, well, I had a question about chitin. I mean, it's a tough polysaccharide. So, and you rightly point out that it's not the only thing in exoskeletons, but I'm wondering, besides bacteria and I guess fungi, is there anything that eats it? And is it useful for soil? I've gotten that question a lot recently because we had this huge cicada, periodical cicada situation here in Maryland, and people were asking about that.
Yeah, I don't know. I don't know exactly what decomposes it or what effects it might have. We've done some soil decomposition experiments where we put prey remains from spiders on the soil and look at how it affects soil decomposition. And there appears to be an increase in soil respiration when you put the exoskeleton on the soil, but I don't know if that's just a little bit of protein and lipid left in the prey remains or some silk wrapping up the prey remains, or if the microbes are starting to decompose the exoskeleton right away. I saw one study where they looked at decomposition of exoskeleton and they buried exoskeleton from some arthropods in soil, and they came back a year later and they could still identify pieces of exoskeleton. So,
and whereabouts was that?
Sorry?
Whereabouts was that where the exoskeleton was buried and it was still recognizable a year later?
I can't remember. I can try to look up that paper.
Oh, Yale Lubin says, isn't this what Dror Helena's group is doing? So I don't know if that rings a bell.
Yeah. Dror and I have a grant together to look at some of the ecosystem effects of spiders and decomposition. And so we've been looking at nitrogen cycling, and he's done some really exciting experiments looking at how white widow spiders in the desert affect nitrogen cycling underneath shrubs in which the spiders live. So, yeah.
I know that I have a biochemist colleague that has bacterial cultures that do eat different kinds of polysaccharides, but my understanding is that they formed, mats and were aquatic. So I don't know. Okay, yeah, and Ripstra mentions some fly larvae have a chitinase, so some insects can handle those tough polysaccharides.